Hello, everyone. My name is Sophie Rovner. I'm the Assistant Managing Editor for ACS News and Special Features with CNEN and moderator of today's webinar. Thank you for joining us today. Through CNEN webinars, we hope to deliver critical information to select audiences in a timely and interactive manner. Although the format may vary, the webinars share a few things in common. First, each webinar comes to you through funding by a sponsor. CNN works with the sponsors to identify topics of interest and value to our audience that are consistent with CNEN's mission. In all cases, a webinar topic is one that CNN would cover or is already covering in its news gathering operations. Second, each webinar is archived at CNN Online and at the sponsor's website after the live webcast. This way, those who can't join today can access the information and discussion at a later time. During the webinar, you can adjust the size of the slides on your screen by grabbing the lower right corner with your mouse. If you need technical assistance, please click on the Help widget at the bottom of the screen or type your query into the Q&A box. If you're disconnected during the webcast, please log in again according to the instructions you received earlier. Each webinar success depends on your participation. Please feel free to ask questions at any time during the presentation through the Q&A box on your screen. The questions will be answered at the end of the presentation, and as your moderator, I will pose as many as time permits. Lastly, CNN does not endorse any company's products or services that may be mentioned in the webinar. Having said that, I'm delighted to welcome you to today's webinar, which is being sponsored by Novacep, which operates 12 R&D and manufacturing sites worldwide, including 6 FDA-inspected manufacturing plants and 1 SafeBridge-certified HPAPI plant with approximately 1,200 employees. Novacep masters key manufacturing technologies, combining purification and synthesis with special capabilities in hazardous chemistry, asymmetric synthesis, HPAPIs, cryogenic reactions, biocatalysis, process chromatography, and membrane filtration. Today's webinar will be presented by 2 speakers. Dr. Jin Suk Hur is R&D and Pilot Lab Manager for Novacep. He joined Novacep in 2006 as a process development scientist and became R&D project manager in 2008. Currently, he is in charge of the R&D and pilot labs at Novacep Process in Pompeii, France. His research focuses on separation processes with particular emphasis in preparative chromatography. He received a PhD in chemical engineering from Purdue University. Our second speaker is Dr. Jeff Marcoux, who is Technical Business Development Manager at Novacep. He joined Novacep in 2005 as head of a process R&D unit in Chasse-sur-Rhône near Lyon, France. He has also been responsible for several oncology API projects as project manager for the Le Mans, France site. He received a PhD in organic chemistry from Montreal University. I will now hand the webinar over to Jeff.
Thank you very much for the introduction, Sophie. Hello, everyone. I'm Jeff. Today, after a brief overview of the main technologies available to produce chiral molecules at large scale and the current role that chromatography could play from the point of view of process chemistry, we will review the main preparative chromatography techniques, its main principles, the scale-up performance, and the technology it depends upon. The second half of the presentation will cover the industrial applications of chiral chromatography, including the development methodology used for its application at large scale. Several case studies and examples will be given in early and late development phases, as well as in commercial production. The most direct source of chirality in chemistry is coming from nature. Chiral starting materials coming from the chiral pool, such as carbohydrates and amino acids, are widely used, but the number of useful molecules is limited in comparison with the increasing complexity of active structures. And often, the extra steps required to functionalize the chiral center are not cost-effective. Enzymatic resolutions and chiral resolutions via diastereoselective salt crystallization remain an extremely useful approach since they are relatively easy to develop at large scale for acidic and basic APIs. Although they can be cost-effective, even at commercial scale, their theoretical yield is limited to 50%, except for DKR, dynamic kinetic resolutions, which are target-specific and are not widely applicable. An important technology consists of creating chiral centers using enantioselective reactions. This area remains an important focus of research, and significant progress has been made in the past 20 years towards the development of widely applicable conditions. Today, asymmetric enantioselective syntheses are routinely used at small scale, but they remain underrepresented at kilogram scale due to specific limitations that we will briefly cover later in the presentation. In addition, the process development phase for these reactions is often tedious and lengthy, partly explaining why they are seldom used at large scale for first-generation processes. Moreover, virtually no cost-effective examples are available for the creation of quaternary centers at commercial scale. Today's topic involves chiral chromatography as an alternative to chiral resolutions and asymmetric synthesis in large-scale production. One objective of this presentation is to introduce the technology to process chemists that are involved in active ingredient manufacturing and process development activities, but that do not necessarily have a broad knowledge of chromatography or have never used it in a development setup before. If, like me, you were trained in academic labs as an organic chemist and have used flash chromatography to purify your compounds, you may be tempted to see chromatography as a sign of weakness in process development. Most organizations have included chromatography as an efficient way to purify and isolate APIs obtained via the medchem route. The quantities involved at that stage are very low, typically at the gram scale, and are typically purified by flash chromatography or one of its many more or less automatic variants using irregular silica gel. Preparative HPLC has been more widely used in the past 5 to 10 years to speed up process research activities and isolate or purify intermediates on small scale, let's say typically less than 1 kilogram scale, to work on downstream chemistry, and also in several cases to speed up the delivery of tox batches. While the use of chromatography has gained wide acceptance amongst process chemists for these early phases of development, we all agree that its cost is very high per kilogram of API produced, and because of that, many do not consider to include it in a commercial process. So at that stage, the chromatography technique and process have no impact whatsoever in reducing the commercial cost of the API. Chromatography can also be used later in the development program to supply GMP material, either for the first chemical supplies or in larger quantities for phase 2 and phase 3 studies. It is not as well known that not only it can be used as an interim solution during the development program, but that it could also be implemented as a result of a route optimization program, that it could turn out to be the method of choice to allow larger scale supply, and we should make the point that it can also be a strategic part of an optimized, validated process for commercial production. In this case figure, the cost of chromatography could be very appealing, depending on the specific nature and behavior of the chemical structure to be chirally purified. And in the favorable case, it can have a tremendously positive impact on the commercial cost of the API. At the same time, this is where the reluctance of process chemists is very high, mainly due to the lack of knowledge about the capabilities and advantages of the different types of industrial chromatography. I will now switch over to Jin, who will cover the technical aspects of chromatography.
All right, thanks, Jeff, and thanks everyone for joining us today. In the first part of the presentation, I'd like to introduce you the chromatography technology and their scale-up and development methodology. And then my colleague Jeff will take over the presentation again for industrial applications and examples. So there are several different types of chromatography depending on the types of interactions among the compound, stationary phase, and mobile phase. So when a mixture of compounds, such as racemine mixture, is introduced to a column, which is packed with the chiral stationary phase, for example, if the compounds have different strengths of interaction to the stationary phase, they will have the different movement inside the column. If one is less retained to the stationary phase, it will move faster than the other one, which is more strongly retained. The retention is affected not only by the stationary phase itself, but also by the eluent strength and polarity too. At the end, we will have the separate products in different fractions thanks to this different retention time. Analytical chiral chromatography is one of the most common analytical tools to identify the compounds and to estimate the optical purity. For this purpose, we should have a good resolution and efficiency for a small diluted injection in general. However, when it becomes a prep chromatography for large-scale production, the injection amount becomes very important matter, even if we have to give up some resolution. It is simply because the injection amount directly affects the overall production cost. So by injecting more in a certain run time, it will be more productive and take less cost. In addition to the overloaded injection, another common technique is so-called stacked injection, especially under a batch mode. When you have an optimum loading amount so that you know that you cannot load more, this stacked injection can increase the productivity by reducing the runtime. If you look at the first chromatogram on top of the slide, you will find the second peak completely elutes in about 14 minutes after the injection. If the next injection is made after the elution of the second peak, the runtime would be simply 14 minutes. But you may already notice that there is nothing happened during the first 7 to 8 minutes after the injections until the first peak starts eluting. So in order to use the stationary phase more efficiently, we can think about injecting the feed every 6 to 7 minutes so that we can have the first eluting peak just after the second eluting peak from the previous injections. By having this simple recipe in your system, you can reduce the runtime to less than 50% compared to original condition of 14 minutes, and of course this will double the productivity. In general, the first chromatography development can be performed using an analytical size column, typically 4.6 millimeter internal diameter and 25 centimeter bed length. After, you may need a much bigger column for your productions. For example, if a process was developed using 2 millimeter ID column, and then it needs to be scaled up to 200 millimeter ID column, then the scaling factor is simply 10,000, which means you have a 10,000 times more stationary phase in your column, and you have to handle 10,000 times more and faster eluents, and 10,000 times more of your feeds need to be injected. Two key questions about this scaling might be how the flow is well distributed inside the column, and also how the stationary phase is well packed in large diameter column so that it keeps the uniform movement of the elution inside the column. As a system supplier, Novasep spent quite a long time and resources to investigate the optimal design of a prep column in order to secure the good flow distribution. As you can see in slide 11, if it is not well distributed, you will see the profile in the left picture, for example. With this flow distribution, you will find a big shoulder eluting before the major part of the peak, or sometimes a tailing, which will contaminate the product or significantly reduce the recovery. In the other picture, however, you will find a well-distributed elution profile. Another important technical aspect for scale-up is how to pack the stationary phase with no channeling but with only good efficiency. For bench scale development, some of you may use a pre-packed column with more than 40,000 plates per meter of column efficiency using, for example, 10 micron packing. Then you may have a question if you can get the same or at least similar efficiency on a 1 meter or 2 meter ID column for commercial production. There must be several technical aspects to be considered, including the design of a flow distribution and so on. But the dynamic axial compression is certainly a big help to have and to keep the good integrity of the packing. First, this compression would allow the packing beads to be well arranged inside the column when they are packed in slurry foam. I will show you a couple of pictures about this in the next slide. Secondly, it will readjust the height of the piston to remove any void volume, which can be created several days after starting your production campaign. In addition, it will also allow you to unpack relatively easy by simply pushing down the stationary face out of the column, as you can see in the slide. So here is the picture comparing the arrangement of stationary face beads under dynamic axial compression versus by gravitational sedimentation. You can see very well organized 3D matrix of beads when the dynamic axial compression was applied on the packing. So we have many industrial examples showing perfect linear scale up from analytical size to prep size HPLC column. The slide presents one good example of scaling up that we made from 4.6 millimeter to 800 millimeter internal diameter columns. The scaling factor was about 30,000, and you can see there was no difference in the separation on both small size and 800 millimeter ID column. So now you may notice that the scaling up of chromatography is not something unclear or unrealistic, but it's quite easy and straightforward if you have a well-packed column and also a reliable system. Then the next topic is what kind of chromatography technologies you can choose for your applications. Basically, there are two major chromatographic processes: single column batch, which is the most common process, and multi-column continuous process. A batch process is to separate compounds mainly depending on the selectivity and the resolution. It requires only one column and one pump, and the development and scale up are relatively simple compared to multi-column process. However, you have to have a good resolution in order to obtain both high purity and recovery at the same time. Another separation technology is MCC, which is multi-column continuous chromatographic process, such as a simulated moving bed and vertical. In the MCC, you do not need a baseline separation, which means you can have both high purity and recovery for a relatively difficult separation with a very small resolution. Or when you have a good resolution, you can inject much more compared to the batch process for still both high purity and recovery. Although it is a bit more complex than batch process because of multiple columns and multiple flow rates involved, you can expect a higher productivity and also less solvent consumption in general in most of binary separation cases. Regarding the selection of the processes, it is purely up to your application and short and long-term goals of your production. Again, the big advantage of a prep batch HPLC might be that it is easy to understand how it works because its philosophy is the same as an analytical HPLC. Another benefit might be the versatility of the system. You can use the batch system not only for binary chiral separation, but also for multi-component purifications when you have more than 2 chiral centers on your molecule or some non-racemic impurities to be removed. So it would be an ideal choice, especially for early development stage when you need a purification process as soon as possible in order to produce hundreds to few kilo quantity for your preclinical test and tox test. Of course, it could be also used for commercial production in a later stage too. In the slide, you can find the pictures of a pilot and commercial scale batch system. Another well-known batch process is a supercritical fluid chromatography, SFC. The separation performance is the same as the batch HPLC process. The only difference is that the CO2 is used for the diluent with some organic solvents. The fast mass transfer under SFC mode allows you to run the system at a higher flow rate than HPLC, so that you can expect a shorter run time for each injection, which will increase the productivity too. In addition, since CO2 will be vaporized at the outlet, the product would be concentrated in the cost solvents, which will reduce the cost for isolation after chromatographic separation. NovaSep SFC system also has a built-in CO2 recycling unit, so the majority of CO2 is recycled back to the system. This SFC could be an ideal option, especially for early development stage. The next chromatographic process is MCC. It is well known as SMB, and NovaSep has also introduced and commercialized an advanced SMB, which we call the Vertical. The system is equipped with a several short column instead of long column for batch process, so that you have a more degree of freedom to play with. Again, since it does not need baseline separations, you can introduce more of your raceming mixture into the same amount of stationary phase for a batch process. And this will allow better using the stationary phase for a higher productivity, typically 2 to 3 times, or sometimes we achieve the 10 times better than batch process. The next slide is a short animation of a vertical process to show you how it works. There are 2 inlets, feed and diluent, and 2 outlets, the raffinate and extract. The raffinate in red is the less retained enantiomer, and the extract in blue is the strongly retained enantiomer, for example. Since the internal profile moves in the same direction of the diluent flow, the location of the ports, inlet and outlet ports, are switched to the same direction to the diluent flow in order to keep collecting the pure parts of extract and raffinate profile. In traditional SMB, the inlet and outlet ports are switched at the same time, which we call the synchronous switching. But in vertical process, as you can see in the animation, they are switched in different times. And this allows us to more precisely distribute the number of columns for each zone. In general, approximately 20 to 30% better productivity could be achieved by vertical compared to SMB process. Between batch and MCC, there is another process, it's a cyclojet. In terms of hardware, the cyclojet needs only 1 column, just like batch process. But in terms of separation performance, it is very similar to MCC. For example, let's assume that we have a quite difficult separation as shown in the slide 20. Because of the poor resolution and selectivity, it is not possible to obtain both high purity and recovery at the same time under a batch mode. In this case, you can certainly use the vertical process, but in addition to vertical, now you can use also the cyclojet process too. The main idea of cyclojet process is to have the overlapped part in the middle of 2 peaks reintroduced to the column inlet in order to use the stationary phase again. So if you look at the separation profile on cyclojet in the right bottom of the slide, the first step is to collect the less retained product, which is raffinate in blue, and then the overlaid zone will be recycled back to the column inlet. During this recycling step, the feed injection is also made, and there is no input or output, so that there will be no eluent consumption too. Then the more retained compound, extract in red color, will be collected in the third step. So we have gone through the chromatography, some principles and some technologies that are often used for chiral separations. Now we can move to the industrial applications and examples about how we develop and scale up a chiral chromatography process in different development stages from lab scale to commercial scale. The graph in the slide shows a kind of rule of thumb about selection of chromatography technology in different development stages and different scale. Of course, there would be an optimal choice for your specific needs and applications. Development of the chromatography process is somewhat similar to development of chemical process. We start with a small analytical scale to screen and to optimize the process. When a promising condition is found and a development work is completed at a bench scale, and the first scale up could be made in 100 to few kilo scale. After the process is finalized and confirmed at a pilot scale, you can work with a custom manufacturing organization, CMO, or you can buy a system for your in-house production. Of course, Novacep can provide both services for you, as well as process development and at bench or pilot scale. In the screening stage, various different chiral stationary phase and mobile phases would be evaluated in order to find the best combination of stationary and mobile phase for your separation. After screening, each operating parameter would be optimized for high productivity and good process robustness in order to minimize any risk during the scaling up. Most of the development work at this stage could be performed using an analytical size column. However, it is very important to use a prep stationary phase, typically more than 10 micron particle size, so that you can use the bulk phase, exactly same bulk phase for your production in the later stage. After, the next step is a pilot scale study to prove the design concept obtained at the bench scale. And this is the first scale up, and the operating condition would be fine-tuned at this stage. Since the pilot study is performed on a small but still commercial scale unit using 5 to 8 centimeter ID columns, you can be quite sure about scaling up after this proof of concept step. So we are now ready to move to the commercial scale HPLC system, more than 1 meter ID column. And this is for my part, and Jean-Francois will take over the presentation. Thank you.
Thank you, Jin. So we will now present a few examples on the use of chiral chromatography in early development. Our first early development example is shown here. In this case, a chiral amine was reacted in step 1 to form a ring structure in 85 to 92% isomeric ratio at the position alpha to the ketone. At the second step, the ketone was selectively deprotonated at the alpha position using an excess of a chiral base at low temperature and was reacted with an alkyl halide to give, after a purification step, the desired enantioenriched quaternary center in 45% overall yield from the amine. The main drawbacks of this approach are the need for formation of a salt break, and crystallization to reach the desired chiral purity. Also, there is a use of an excess of the chiral base, which ended up being the main cost contributor to this chemistry. And despite a possible recycling of the base, actually the chiral base accounted for about half of the cost of all raw materials, since 7 kilos of base were required in the reaction per kilo of substrate to convert. Going up in scale was problematic also due to the limited availability of the chiral base. A chiral chromatography process was proposed based on the preparation of a 1-to-1 mixture of diastereomers, starting with the same chiral amine. In this case, since no chiral base or diastereo selectivity is required, cryogenic conditions are not needed, and a simple one-pot reaction conditions were defined. A chromatography screening was performed, and acceptable conditions were found for the separation of the two diastereomers on chiral stationary phase to give good stereomeric purity in a comparable overall yield of 40%. The chromatography conditions resulted in milder alkylation conditions without the need for a chiral base and low temperature reactors. Although the aperamization and recycling of the undesired isomer was not possible, the projected cost of the API was reduced by more than half and led to an easier scale-up to the 100 to 200 kilogram range. The development of the active was discontinued for other reasons, but the chromatography approach was seriously considered for future deliveries. A second example concerns the synthesis of a chiral aromatic amino acid in support of phase 1 studies. The chirality is introduced in an elegant fashion at the last step of the synthesis via an asymmetric hydrogenation of the N-amine, or aminoinoate precursor. The precursor to the hydrogenation was obtained by a palladium-catalyzed Heck coupling reaction between an aryl halide and an aminoinoate derived from serine in 3 steps. While the enantiomer selective hydrogenation conditions using a chiral ligand complexing the ruthenium-based metal catalysts offered very good stereo control at more than 98% EE purity on multigram scale, several drawbacks resulted from this route. The use of transition metals in the last steps of the synthesis raised the potential issue of residual metals in the API, which may require specific treatment steps. In addition, traces of palladium from the Heck coupling needed to be removed prior to running the asymmetric step in order to get good enantiomer selectivities. A major limitation was the use of an expensive asymmetric catalyst, which was only available from a single supplier with long lead time and lack of control over its availability. The last drawback is very challenging. To lower the cost contribution of the catalyst, one needs to minimize its charge. Poisoning of the catalyst resulting in low catalyst turnover becomes a huge issue if the starting material is not highly pure. One choice is to increase the catalyst loading, thereby increasing its cost contribution to the API. Another choice is to design a process that gives a very pure hydrogenation substrate, sometimes even requiring purification by chromatography. The current process was limited in size due to the availability of the chiral ligand and suffered from long lead times. These scale-up challenges are not unique to this project, and some of the most common limitations to asymmetric hydrogenations are outlined in here. Often, the most important cost contributions do not come from operations in the workshop, but from the cost of the catalyst. Part of the cost comes from the high commercial value of the transition metal itself, typically rhodium, ruthenium, or in some cases iridium. And another important part comes from the chiral ligand. Many have a complex structure, which is not that simple to synthesize, and intellectual property rights on many ligands add to the cost in the form of royalties with single sources and limited availability. Consequently, acceptable costs at commercial scale require very low catalyst loadings, typically 0.1 to 0.2 mole % in many cases, which is a challenge by itself for process chemists. Impurities that could poison the catalyst need to be tracked and eliminated, and a purification step of the precursor is often required, adding to the cost of the process. Asymmetric hydrogenations, while elegant and powerful, require significant efforts and time to reach cost-effective conditions, which may not be compatible with the available timeline to provide GMP bulk in support of clinical trials. In general, chiral chromatography can be either an interim or even a long-term alternative to enantiomer selective synthesis. An alternative process was proposed in this case based on the preparation of the racemic amino acid in 3 steps, starting with the corresponding aldehyde via a Stryker reaction. The racemate can then be separated by continuous chiral chromatography at step 4. This route offered the advantage of more straightforward chemistry and acceptable productivity of 1.2 kilogram of enantiomer per kilo of a widely available chiral stationary phase per day, with possible epimerization and recycling of the undesired enantiomer. It also offered the advantage of allowing the on-site treatment of most waste streams, including aqueous phase, since the process did not rely on the use of heavy metals. The lead time is greatly reduced by not relying on the chiral ligand, thereby removing the scale limitation. And despite the unoptimized process, it offered a good interim solution until a better process was defined. Let's now switch to the use of chromatography in late development with a first example that illustrates the use of chromatography for the preparation of commercial batches. Our modafinil, which is the R enantiomer of racemic modafinil, is developed and marketed by Cephalon. During the early stage development, it was discovered that modafinic acid exists as a true eutectic mixture or a conglomerate. This conglomerate was separated into its corresponding enantiomers by preferential crystallization. Although several kilos were prepared via this preferential crystallization, this process was labor-intensive and limited in terms of scale-up, leading to unreproducible results. A preparative chiral chromatography approach was used for clinical supply and up to the registration of the API. Starting from scratch, the feasibility screening and in-depth development studies were completed in 4 months. 60 kilos were produced by batch HPLC in 3 months, while a continuous process was being developed. 540 kilos were produced by multi-column chromatography for phase 2 and phase 3 clinical studies over 8 months. And the resulting process was validated at 3 different commercial production sites. In this case, chiral chromatography was used to greatly shorten the time to market. Even if the ultimate cost of good objectives could not be achieved with this technology, it allowed enough time for the development of the final process based on the asymmetric oxidation of a thioether, shown below as a third strategy, which was filed as a post-approval change. The whole process from screening to validation was completed in just over 23 months. And this slide shows the usefulness of preparative HPLC as a faster development tool and reasonable cost to provide phase 1 and 2 supply, followed by continuous chromatography as a reliable option for commercial production that drastically reduced time to market. Chiral chromatography could also be a tool to consider for agrochemicals in certain cases. Here is one example of a screening and optimization study performed for an agrochemical active, which was initially developed as a racemate, but where one enantiomer showed undesired properties and activity. There was a need to quickly define an access to the desired enantiomer, as the potential application of the ingredient was limited in time. Process development work was conducted, as previously described by Jin, and several chiral stationary phases were studied. The initial screening was performed on the active itself, but also on the intermediate products during the synthesis, and further screening was performed on related derivatives in order to identify several possible solutions. Productivity figures for the active ingredients were high at 6 kilograms of feed per kilogram of phase per day, but without the possibility of racemizing or recycling the undesired enantiomer. On derivatized precursors, productivity figures went over 10 kkd in certain cases, which is considered a high productivity. To give a cost indication, depending on the precursor and the productivity, a cost range of $15 to $60 per kilo was obtained at 100 to 1,000 metric ton output per year. This cost for this separation includes the cost of the chiral stationary phase, amortization of the chromatography unit, labor, energy, and raw material costs. For high productivity separation, the cost can be lower and much lower in some cases than classical resolution, especially in cases where the recycling of the undesired enantiomer is not possible or is possible for chromatography. the chromatography approach can be cost competitive at commercial scale. As a chemist, I consider that chromatography has a definite edge over organic synthesis for the reliability of the cost evaluation. And unlike chemical steps, scale-up of chromatography is linear, so screening results provide reliable cost and productivity figures at very large scale. Another example briefly outlined here is retafaxine, developed by GSK. The initial route relies on a nice kinetic dynamic resolution on large scale, with 260 kilograms of input material using only 194 kilos of solvent per kilogram of product. Despite its efficiency, the Chiral chromatography evaluation indicated that it could be more cost-efficient overall and had a tremendous impact on the projected reduction of overall waste load by more than 50 metric tons a year at commercial scale. This process received the U.S. Presidential Green Chemistry Prize in 2005 and won the AstraZeneca Green Chemistry Prize also in 2004. The last part of the presentation is devoted to two examples of commercial processes based on variable multi-column chromatography technology. Sertraline is marketed by Pfizer, and the initial synthesis reported was based on the preparation of racemic tetralone as a key intermediate prepared in 5 steps and 31% overall yield. The ketone was then reacted with methylamine via Lewis acid catalyzed reductive amination to give racemic sertraline, which was then resolved with mandelic acid in 78% of the theoretical yield. The main drawbacks of the initial process was the chiral resolution, which occurred at a late phase in the synthesis, with significant loss of expensive product due to limited yield and the fact that no recycling of the undesired enantiomer was possible. High costs were associated with the diisomeric salt resolution due to the extra steps of the salt formation, salt break, and the resulting chemical waste. The commercial route shown here was based on an improved preparation of the racemic tetralone from the starting naphthol in a single step via a Friedel-Crafts arylation. The racemate was separated by multi-column chromatography, which was then submitted to reductive amination. Chromatography conditions resulted in high productivity, a high yield with more than 45% of the desired enantiomer, translating to 90% of the maximum theoretical yield, in high purity of more than 99%, and the undesired enantiomer can be racemized and recycled. Very significant cost savings were obtained: capacity, labor, and raw material cost savings. And the costs were in line with the best examples developed in the pharma industry. As Pfizer reports it, although enantioselective syntheses of the required S enantiomer of tetralone have been achieved, none of them has been commercially viable. On the other hand, the improvement on the synthesis of racemic tetralone was so successful that with this simple industrial procedure, the late introduction of chirality has been possible without an acceptable cost penalty. The last example is Keppra, UCB's top-selling drug produced by chiral chromatography. The chromatography developed was very cost-effective, with a very high solvent recycling rate, using only 0.2 volume or 130 ml of solvent per kilogram of pure enantiomer produced, with an environmental impact for the global process being reduced by 89% versus the alternative process based on diastereoselective salt formation. In a nutshell, the chromatography can make it to the market when a good productivity is demonstrated on commercial CSPs, chiral stationary phases, and the racemic compound has the right physical properties. It cannot replace good chemistry, and an efficient process involving chromatography needs to rely on good chemistry. And currently, several late-stage compounds are being chromatographed at ton to several hundred-ton scale in 2012 in the industry. So we hope we were able to convince you that chromatography is not a sign of weakness in process development. It is GMP, very predictable, and gives reproducible quality profile. It is solvent economical at large scale and may turn out to be very cheap. So it is for medicinal, but also for process chemists. And it is a fact that chromatography is a real production tool with low cost potential. Process chemists should consider chromatography screening in their development activity. And Chiral chromatography should be systematically evaluated for large-scale API manufacturing via a simple screen to find out whether this could be useful towards the identification of the optimal commercial process. Thank you very much for your attention. So Jin and I are available to answer any question you may have submitted during the presentation.
I'll also add that listeners can continue submitting questions until the end of the webinar, and the speakers will follow up with you later if they're not able to answer your question today. The first question is directed to Jin. How often do preparative columns need to be repacked?
The lifetime of stationary phase, there are two aspects to be considered. First one is the physical stability, and the other one is the chemical stability. Physical stability is more related to the operating pressure because you have to stop pumping for injection and start. And during the operation, there will be some variation in the operating parameter, pressure, temperature, and so on. Usually, we use the stationary phase that has a good record of this physical stability. So we are not that concerning about this physical stability. However, the chemical stability is more difficult to predict because it's very, very related to the quality of crude material. So during the development study, we usually do the lifetime study. So we introduce the crude material to the stationary phase and regularly check out the efficiency and the separation performance to estimate how long we can use the stationary phase. So the answer is that this chemical stability, we have to evaluate the stability based upon the crudes that you want to purify.
Next question directed to Jeff. Can the speakers comment on the viability of industrial scale chiral chromatography as the last step of isolating an API? The concern is that even if you can recycle the undesired enantiomer, haven't you added additional GMP processes?
This is a good question. It is actually the case, depending on the strategy that we're using, that an additional GMP process might be added as a result of the racemization process. We have different approaches possible. The first one is to actually separate the racemic material and then file an additional chemical step for the racemization and the chromatography of the racemic material coming from the racemization. A better approach when it can be designed is to fully exploit the continuous aspect of chromatography. Continuous chromatography by design reaches a steady state where basically the feed of the racemic compound can be tailored to include the feed from the racemic material obtained from the synthesis with a portion of the feed coming from the racemization process. If you can show steady state in that kind of mixture with some appropriate ranges defined for robustness studies, then you can actually include the racemization process as part of your final chromatography process. And we have had experience on these things, knowing that all of these solvents can be recycled from the chromatography aspect and also from the racemization process, which can be run in a continuous fashion in parallel. A third approach is also to have in your reaction conditions of the racemate out of your synthesis conditions that will epimerize anyway the racemic compound before chromatography. So the undesired enantiomer can just be basically recycled at the N-1 step of the GMP process. and this is in this case not considered as an additional GMP step.
What are typical operating conditions for the chromatography operation? That's directed at Jin.
Yes, for the bench work, we typically use a 4.6 millimeter ID column with 25 centimeter bed length. For this column, we use about 1 ml per minute flow rate at 30 degrees C. That's just the test condition that we are doing. And after we can change the flow rate, also we can change the temperature if it is not appropriate. But I should say that 1 ml per minute and 30 degrees C might be the reference conditions. And then we can go from there to optimize the process.
For Jeff, what are the U.S. and Europe regulatory requirements / guidelines for use of chromatography in chiral separations?
This is a difficult question to answer in a few sentences. Let's say that the requirements and guidelines are much in line with what you would expect for chemical reactions. You need to validate the process. You need to show robustness. In the case of the chromatography, we have additional help since the inline following of the separation, either by UV or by other techniques, is the perfect and ideal PAT type follow-up that are more difficult actually to implement on chemical steps. So also the continuous chromatography, by definition, since it's running, it's reaching a steady state, you can monitor at each time the behavior of your system, and basically the definition of the process is fulfilling the regulatory requirements. Also, I must say that this continuous chromatography, or also the batch chromatography, the way Novacep is running it, is actually using only one fraction for the hard fraction in batch chromatography. I think we're running into potential GMP issues when chromatography is run by taking several fractions that are analyzed separately and reunited. But the beauty of the batch HPLC chromatography is that we can define a hard fraction, a single fraction, which is then analyzed for purity, passing IPC requirements, purity requirements, and the process is very reproducible, and we just have to show the reproducibility of the purity profile, and we can actually stack up basically several injections. The continuous chromatography, by the same definition, is not running into the problems you might have with more traditional flash-type chromatography.
Question for Jin. The questioner is asking about solvent recirculation. What is the guarantee of recirculating solvent purity, and how many times can we use the same solvent?
So regarding the solvent recycling, during the development study, we check out if there is a specific interaction between the molecule and solvent. Most of case, we control this carryover during the evaporation at less than 10 ppm or even 5 ppm level. But sometimes we found that there is a specific interaction, so we have more than 100 ppm always coming out during the evaporations. In that case, we have to sacrifice some solvent by controlling the operating condition of evaporations so that we can reduce the amount of carryover during the evaporation. So most of the case, we don't see any big problem because of this carryover during the isolation, but it's one of the topics that we are investigating during the development study.
Question for Jeff. Are there any concerns on leachables from columns or from cross-contamination from previous batches?
We can see it in two aspects. from previous batches of the same compound. There is from a GMP perspective, since the continuous chromatography is run continuously, we have a definition of batch of racemic material that enters. So you need actually to define your specifications to show equivalency of each batch of racemic mixtures that enter into the chromatography process. That said, the binary type separation that we're getting from continuous chromatography is designed to separate the two compounds. So basically, it is not necessarily removing minute impurities that are present in the racemate. For example, we can use continuous chromatography for early phase programs and still keep many of the representative impurities coming from the process for toxicology studies. In terms of cross-contamination, when a chromatography unit is used for separation, this is used over a long period of time. So there's a cleaning process that goes on, but typically all the movable parts, such as the filters and basically the top of the columns and many parts are compound specific. So we're not running into the problem of demonstrating cleanliness and cross-contamination from other types of compounds. The same thing also for the chiral stationary phase. By GMP definition, it is assigned to one intermediate of one compound, and each stationary phase is dedicated to the process.
All right. Last question. This is directed at Jeff. The question is how to validate the chromatographic operation. And second part is asking about the cleaning process. Does that use a large amount of cleaning solvent?
On some of the previous question, I touched on the requirements for the validation of a chromatographic operation. So I will mainly focus on the cleaning operations, which require a lot of solvent. Actually, there's of course a cleaning at the initiation of the process, but a continuous chromatography process needs to be designed to run over several weeks and even months, with many batches run over the same chiral stationary phase. We need to be very careful about anything that can build up in the chromatography system. But that's why solubility studies need to be done and make sure that the eluent will actually solubilize all organic impurities. So since it's a steady state process, everything that enters comes out at a certain point, either in the raffinate or in the other solvent streams. The eluent is then recycled via distillation, which is then analyzed for the lack of organic pollutants. But the distillation is a nice way to actually use these. What we have to really worry about are inorganic impurities, buildup of salts, and we need to define a specific filtration system and also pre-treatment systems in line with cartridges to make sure that this aspect will avoid pressure buildup over days and weeks in the chromatography system.
I'd like to thank our audience for their stimulating questions and Dr. Her and Dr. Marcoux for their engaging presentation. Thank you On24 for technology and production services, and thank you Novacep for the sponsorship that made this interactive webcast possible. Be sure to check CNN or CNN Online for information about the next edition of CNN webinars. For CNN webinars, I'm Sophie Rovner. Thank you.